Okay, I think we just got the go-ahead. So I think we're going to start. I'll start by introducing our first speaker, which is Jeremy Forrest. He goes by he, him, and he is working on open source tools for optical imaging. He's currently a postdoc at NYU in the Reyes lab. And when you're ready, Jeremy, take it away.
Hey, can everyone see the screens? Okay, all good, thanks. So today I'm going to talk to you about the software I'm developing, which allows for whole optical electrophysiology. So I'm working in the New York University in the Reyes lab. Basically, what is whole optical electrophysiology? Well, it's the ability to record and stimulate neurons with light. So first is controlling brain activity with light, and that's using optogenetic. For those that might not be aware, it's the ability we have to transduce cells with channels that respond to light. And when the channel responds to light, it opens, leading to activation or inhibition of the cell. And here on the right, you can see when the light in blue here is turning on, we have a spike happening in the cell. Same thing at faster rate. And it's not now routinely used in labs. In our lab, we can use it to stimulate here 36 neurons at random times. And then we can, at the same time, here record for 3 different neurons using standard classical electrophysiology. This has 2 major drawbacks, though, that first of all, it's invasive. So doing patching kill the cell after a short time, and also having a very limited bandwidth in the sense that you would be lucky to do 12 to 16 parallel recordings of cells in the same preparation. So to try and address that problem, scientists have been looking at imaging brain activity with light, which is optical imaging. And here on the left, you can see one of those proteins, which is quasar2, which is sensitive to voltage. And basically, when there's a change in voltage, that protein change in fluorescence. And you can measure that change in fluorescence. And as you can see on the right, quasar2 and standard voltage recording techniques are extremely similar. And you can detect action potential and EPSP that way. So if you combine the two, you have optical recording and optical stimulation. And as you can see on the left, the two, so on the red, you have the traces of optical imaging, and on black, you have traces of classical electrophysiology. And as you can see, they look perfectly similar. So you can, in that way, record with optics, record many more cells at once, and also not invasively. So in theory, you should be able to keep your cell alive much longer. That has a limitation, and among them is that optical electrophysiology requires multiple pieces of equipment to work. Indeed, you need a high frame rate camera to capture the change in voltage, at least at 500 Hertz. You need a laser for the voltage report of visualization. So you need to shine quite a powerful laser on the preparation. You need a digital light processor for the pattern optogenetic stimulation, a microscope with movable stage if you want to move around the preparation. And also, if you want to do that, you need classical electrophysiological equipment. So for that, we developed that software. And we developed it using Python because Python is a rich ecosystem for software development and scientific computing. It is a large and expanding user base, and it's also free and open source, allowing for the sharing and modification of that code. We took a model view control approach, which means that every component in the model, control, and view are completely independent. And by that, it means that the user interface is independent from the data storage component and also independent from the hardware component, so the low-level code. And to take an example for that, in the view level, if I take the camera as an example, a user will be able to adjust the exposition, for example, of the camera, or adjust the focal view of the camera. Then at the controller level, it means that that level is going to store the image data and also the timing of the camera recordings, for example. And then at the model view, the hardware, it's a hardware low-level interface. And basically, it means that if you want to change the view, so the user view because you find it ugly or something else, then you will be able to do that without changing anything else. For the controller, if you want to change the data format your data is saved in, then you can do that without changing anything else. And same thing for the model view. If you want to add some different hardware, then you can do that without changing anything else. Here's a brief overview. So when running the software, you're going to have that main window. And then you can initialize an experiment that's going to generate some metadata file, the timings of the hardware, the array position of the simulation, things like that. Then you can load different modules that are all independent. And for example, microscope, laser, digital light processor, and camera. And overshadowing, or then you can also have some config file modification if you want, and some basic analysis. And then overshadowing all of that, you have an automation module, which would allow for around-the-clock whole optical electrophysiology, which is the purpose of that, the end game purpose of that software. And with that, I'd like to thank my advisor, Alex Wright, and NYU, and the funding sources, DARPA. Thank you very much. If you have any question, please go ahead. I stop sharing for that.
Thanks, Jeremy. Is there anything that you would like to share about your work on this project that you most?
Yeah, basically the purpose of sharing that software that's not quite finished is are people interested in it, and how robust should I make it to user base, to a certain user base. So if I'm the only one using it, then I can make the code that's robust because I know what I'm doing with it. I know where the bugs are. I can go around them. It's fine. If someone else not familiar with the code using it, then it needs to be a lot more robust. And the basic idea is to get an idea of how large a user base would be, if there is any, and then take that into account to then go forward with how I'm coding it. And I mean, opening it up, having people code it, code some stuff, that would be great too.
I see a question from Sophia, so I'm going to relate it to you. You probably can read it yourself too. So Sophia is asking, what sort of feedback do you hope for the community if they wish to use it? What kind of feedback do you hope to get?
Well, like how easy is it to understand, to use, how, if people want to collaborate on code, adding some hardware pieces in it, how intuitive it is in coding, in using it, what's the functions that I'm missing. I haven't been doing electrophysiology for a very long time, so I'm probably missing quite a bit on that front. So things like that. Basically, yeah, user or people that would be interested in using it, having the say in whether we want to see.
So Phil joined us with his camera on because he also has a question.
Mm-hmm.
Yes, Jeremy, first of all, I loved your talk. And as a fellow optical imaging person, I really love when people talk about what we're doing. But so I just noticed immediately looking behind you, like I see, stimulators, and I have stimulators as well that are analog. And so often like A to D conversion can often cause all kinds of problems. So like how robust is this across the board for like different kinds of data input? Because you mentioned a little bit, if you could go into further detail about like how many file types it can take, and what's it called? I'm blanking on the term for this, but like backwards compatibility with like older ways of taking. Because some of there are people who are still using functioning analog stimulators that are like 15 years old, right? And that can cause all kinds of problems. So I was just wondering.
So most of the optical things is a direct query from the camera, for example, for the images directly into Python. So I have a low-level code that creates the images directly, and then intermediate level that saves the data, and then the user data, the user interface that shows the data. For that, there's basically no link between computer and the cameras directly through I forgot the cable format, but anyway, it's a direct link. Then we also want to compare with classical electrophysiology in quite a few cases, at least for now, because that kind of technique is quite recent. And so for that, I'm able to interface, same with Python, and there's quite a bit of available script on GitHub to interface via acquisition card, mostly any acquisition card. At least that's what we have here. And so I'm going through that to then get the data when it's classical electrophysiological data. That answer your question?
Cool. Yeah, so I am clearly not an optimal imaging person. So I was wondering, because your tool is completely open source and yeah, working in the Python environment, is it the case that the optical images are also openly available, so that yeah, if someone would be familiar with your toolbox, they could analyze images that exist in the world? Or
the idea is to make it as open as possible. So there's no data per se going with the software, but I'm hoping to get the data I'm trying to acquire right now. I'm hoping that at one point I can transform it into an NWB standard format. So for those that are not aware, it's a format that is that aims at being completely open source with a lot of metadata in it and completely reproducible. They had a couple of workshops in the past year, which was really interesting, and the idea would be to have that at one point. I'm not there yet because the format is not compatible with that new kind of data. So it needs to be expanded, which is something you can do because their format is completely open source. And there's quite a big community around it, which is great.
Yeah, just for the people that are listening that don't know what NWB is, it's Neurons Without Borders. And yeah, I've played around with that format a little bit as well with EFIS data. Especially because Sophia mentioned earlier the backwards compatibility. As NWB is developing, I sometimes get stuck into some hiccups there, but I think it's a really great and cool initiative, and it's really nice to hear that you're actively working on software tools. Unfortunately, my phone is going to ding in 5 seconds because it's time for us to move on to Phil. But thank you so much, Jeremy. That was really interesting. Sorry. Sophia also posted